Welcome to Brain Ponderings, your window into the world of neuroscience research, conversations with scientists at forefront of their particular subdiscipline in the big field of neuroscience. And today it's my great pleasure to welcome Professor Marc Freeman. He's director and senior scientist at the Vollum Institute, which is at the Oregon Health Sciences University in Portland. He is an expert on developmental neurobiology, neuroplasticity, and particularly have focused a lot on glial cells, those other cells in the brain and nervous system.
Mhm.
And today I'm going to talk with him about astrocytes, which, at least in the human brain, it looks like there's more of these astrocytes than there are neurons. There's a more, uh,
Uh,
approximately the same amount anyway.
yeah. I think so. So thank you for having me.
Yeah.
It's great to be here, Mark. I've been looking forward to it. Always happy to talk about astrocytes. And it's funny when I started, um, people would throw out the number 90% of the brain in a mammal was glia. And I think we've settled on a number of like 50-50, half
Yeah.
neurons, half glia. And within that 50% that are glia, about 30% of the total brain cells are probably astrocytes.
Yeah.
But,
Okay.
you would get a different number based on who you asked. My philosophy, especially given the fact that Drosophila, our main experimental model, the larval brain, for example, has something like 180. It's not how many there are, it's how much influence they can exert over circuits. And I think we're starting to learn that's quite a bit. Yeah.
Yeah. Well, it's a wonderful system to look at this for many reasons. Why don't you talk this, some of the audience is kind of broad for this
Sure.
podcast series. So we've had previous episodes on what scientists are using flies.
Mhm.
Um, Talk about some of the big advantages
Sure.
for discovering uh, functions of genes where the function isn't known, of the proteins encoded, and the advantages for studying development. As you know, I did a lot of work related to brain aging and neurodegenerative disorders, so they're valuable there too because they have a relatively short lifespan. You can get feedback quickly.
Yeah. So flies have been a really wonderful model organism for a long time, since the famous fly room at Columbia, started by Thomas Hunt Morgan. And they have really been a powerhouse for, like C. elegans, another really great model organism for picking apart really hard questions in biology. And the way I often explain it to people is we fly people are geneticists, not necessarily biochemists, and every person has their own approach. Biochemist will take a tissue, put it in a blender, chop it up, say, what's it made of? What happens when I add this or that? And we geneticists, we try to break stuff, right? Like, so we do forward genetic screens where we go in and just randomly break genes. And then we look for phenotypes, right? Changes in the organism that are wrong, right? That where something that should normally happen doesn't happen or it happens differently. And then we go in and we clone the gene and we try to understand how is it that the product of that gene, the protein, has led to that event. And one of the most famous ones that when I give lectures, I'll often tell people about is animal body plan, right? There was this very famous screen by Christian Nusslein Volhard and Eric Wieschaus back in the 70s, where the question was, how does an animal lay down its body plan? How does it make a head and a tail and a dorsal and a ventral, right? And so they did this very simple but incredibly powerful screen where they just made random mutations. They focused on ones that caused lethality, assuming that if they were lethal, they're probably going to have a more prominent effect on body plan. And the way they scored body plan was basically to acid wash maggots, right? They took the larval stage and they just washed out all the tissue and they flattened the cuticle. And the cuticle has in the anterior these beautiful mouth hooks, and in the posterior things called a Filtz corp or a breathing organ. On the ventral surface, they have a denticle belt, and on the dorsal surface, there's their naked cuticle. And they simply looked for mutants that changed that, where there were like two heads, or the entire cuticle was, was covered with a denticle belt and looked furry, or there were even mutants that came out that were really shocking. And essentially, they did that screen. It was a famous paper in Nature, and they cloned the vast majority of the genes that lay down the Drosophila animal body plan, like the Hox genes, and a number. And there's several reasons I love that screen. First is that, of course, evolution works as you move further up the evolutionary ladder. Homeodomain molecules were found in other animals, in mouse, for example, and in humans. They essentially work in the same way in terms of how they establish anterior, posterior, dorsal, ventral. The final product is obviously very different. There's major changes along the way. We don't look like a maggot, but the genes that control how a maggot has a head and a tail are the same ones that make it anterior, posterior for us too. And so that showed that you could use really it was called a saturation mutagenesis too. They really went hard. And actually, as I was growing up in the field of Drosophila, most fly meetings, 80% of the posters were on genes that were identified in that screen where their progeny of PIs had gone out and were just working on those and trying to understand animal body plan. And so, yes, they're used for animal body plan, but they're also important in other processes. But a few reasons I really loved that screen were totally unexpected mutants came out, like one called even skipped, right? Where those animals missed every other segment, right? And if you had to try to reverse engineer that and think, why would you design an animal there so there's a gene that makes these segments but not every other one? I never would have predicted that. But like you let the animal tell you how it works, and then you try to deconvolve how that process works. And so that is extremely powerful. And people still do that now, but I have to admit that I wish fly people went back to screens more, because now we've become a little too enamored with these other technologies,
Ah,
which are great, right? Genome engineering is great, reverse genetics are great, but the power of the system is that you could just ask, how does this work? And just start breaking things and let the animal tell you, right? And so that Nusslein Volhard screen was a great example. They were able to do it because flies are inexpensive. You could go from embryo to adult in about 10 to 14 days, depending upon the temperature you raise them at. You can clone the genes and manipulate the genome quite well to figure out how those genes work. And then once you find them, you can use enhancer suppressor screens to find other components. You look for mutants with similar phenotypes. You often found they were in similar pathways. And so it was really just if anyone wants to read a beautiful classic paper on genetics, go read that screen from Nature, from Nusslein, Volhard, and Bischoff. And so people like me, for my career, I started working on fly, working in olfaction in John Carlson's lab at Yale as a grad student, because when I went to grad school, I really liked genetics and molecular biology, and that sort of, pushed all my buttons. I fell in love with it as a model system because I felt like there's so many interesting questions that we still need to answer in biology, and there's lots for flies to still contribute, and worms and yeast and other model genetic organisms. And then I went and did a postdoc with Chris Stowe at the University of Oregon, where I started to study developmental neurobiology. And like you, I became fascinated with that and have sort of continued to do that in my own lab, along with some work that is also in the adult. But there I began to focus on, how a few people had started to use flies to really seriously try to understand glia, but not nearly to the extent that they had been used to really make contributions in things like neuronal development, axon pathfinding, cell fate specification, and all that, right? Where they had been enormously useful. And so Chris, actually, I remember I went into Chris's office and said, Chris, I think I'd like to switch from studying neurons to glia. And his response was, sure, great, go ahead. Just make sure you take everything with you when you leave, and they laughed. And And that's when I switched. And so since then, we have been trying to use Drosophila to understand glial biology. And again, it was here are these cells which first we had to characterize how many different subsets flies had, but then we could use these genetic approaches to ask how do you build them? How do they regulate neurons? How do they help control the assembly of circuits? Once those circuits are assembled, how do they play important roles? And so it's really unlike there's amazing things you can do in mouse now, especially with conditional mutants and so on, but it still remains an enormous challenge to do a forward genetic screen.
Yeah.
And I think there's still just an endless number of really exciting questions people could still answer in flies if they decided to.
What are some of the key mechanisms that have been discovered that determine control stem cells, if you will, the precursors of either neurons or glia? Because
Mhm.
at some point in development, there has to be cells that are probably capable of producing both, and then there's some changes
Yeah.
go on that, if whatever, gene X is turned on or turned off, you get neurons, or if this gene. And is it similar in flies and mammals?
Yeah, exactly. So the answer is yes. It's not exactly the same, but it's one of these things where fundamental principles shine through, right? And this is what got me so excited about going to Chris Doe's lab, right? Because Chris is very well known for studying at least three different things, which are crucial, probably even four, for us to have made progress in that field. So the first is that in the embryo, there forms this highly stereotyped neuroblast map, right? And Chris actually has a thing of it on his website where in every segment, there's a midline and there's a mirror image on either side of stem cells that formed, and they're so highly stereotyped that each neuroblast has a unique name, like NB7273, et cetera. And people like Chris and Gartecnow and others have gone through and done dye labeling. So not only do we know the exact array of neuroblasts, but we also know the full clone of their progeny based on dye labels, both neurons and glia. And so when I joined Chris's lab, he and Garrett and others had just assembled essentially a complete map. And one of the things that's really interesting about that is in the embryo, it is highly invariant, and it's so stereotyped that people in the field would jokingly call it the worm in the fly, because C. elegans is known for having basically invariant lineages. These neuroblasts, not completely, but to a large extent, are somewhat invariant. And Drosophila was used by Chris and others to study things like how do you make those stem cells, right? How do you pattern them along the AP and DV axis? Within each segment, there are 7 rows of neuroblasts, and those first you have to pattern, for example, neuroepithelium. How do you create a midline? How is the patterning done there? The mechanisms that drive that along the DV axis are well known and very well conserved in flies. So there's VND, IND, and MSH are famous transcription factors that are ventral, intermediate, and dorsal in the fly, and there are corresponding transcription factors in mammals in similar domains. Within each segment, there are these patterning genes that will specify each of those rows, 1, 2, 3, 4, 5, 6, 7. And those are the segment polarity genes, which are like hedgehog and wingless and all these other things that feed into forming those fates. And together, those AP and D, the AP and the DV's patterning mechanisms essentially allow each neuroblast to know what it should be. Then that neuroblast gets specified and makes its progeny. So the first major thing was how do you make a neuroepithelium and pattern it? And that was beautiful. And actually, many of those genes came from that Nusslein-Volhard screen, right? That was a screen that pulled out. So it's called hedgehog because the denticle belts aren't segmented anymore. They just run the whole length, right? And so it looks like a hedgehog,
Yeah.
right?
Yeah.
So it's often a really good reason that genes have funny names. And if you're working on a gene with a funny name, it's probably because it was identified in a fly or a worm, right? So that was one thing. Once the
The Sonic Hedgehog is interesting because we played around with a little bit. I had one of the lab members that was really interested. And
Hmm.
so the receptors for this sonic hedgehog, which, as you said, is discovered for its critical roles in patterning
Hmm.
during development, are in differentiated neurons.
Yep.
And in our hands, they're in the dendrites,
Yeah.
but not the axon. But
Okay.
if you activate the sonic hedgehog receptor in the dendrite, the axon grows fast.
Oh, really? Yeah.
Yeah,
Well,
we
yeah.
tried to figure that out. We didn't really. We had a J neuroscience paper and had it up there.
Yeah. Axons and dendrites can talk to each other in really interesting ways.
Yeah,
Um, but yeah, sonic hedgehog is the one of the vertebrate orthologs of hedgehog, right? That's
yeah,
why it was called that. So that was so patterning was one, and then you make those stem cells, and the next thing that happens is they start to produce these progeny. And when these stem cells divide, there are these huge neuroblasts. They divide and make a daughter cell called a ganglion mother cell. And then that daughter cell divides once and makes two cells, neurons and or glia. And that process of asymmetric cell division turns out to be governed by a machinery which is very similar to the cell polarity machinery discovered, like the PAR genes in C. elegans, right? So PAR6, APKC, et cetera. So you have a whole molecular machinery that establishes this is going back now to my postdoc days, but I think I can remember it. Apical domain, which says basically you're bumped up against the epithelium, and a basal domain. And during that division, there is actually a large amount of molecular machinery that Fumio Matsuzaki, Chris, and many other groups. I will not do justice in naming them all, but it was basically found in flies and really worked out beautifully that there's a scaffold that develops the basal machinery, sorry, the apical machinery tells the basal machinery to go to form a crescent where the cell's about to divide. And when the cell pinches off, all this machinery, including, for example, the transcription factor that Chris cloned when he was back in Corey Goodman's lab, Prospero, gets dragged asymmetrically into the daughter cell and tells that cell, you're a GMC, right? Then that GMC divides once and makes cells based on the particular lineage. So the asymmetric cell division machinery in neuroblasts was, in a large part, worked out by people like that, working on the fly stem cells. Then the next thing that happens that's quite amazing is what's called temporal cell fate specification, right? So if you were to look at any given neuroblast, like neuroblast 1-1, and you were to die-eye label it over and over, it doesn't just make a random group of cells, it makes the same orderly progression of cells. So daughter cell 1 is 1, 2 is 2, 3 is 3, and so on. So how a stem cell knows where it is and counting which progeny it's going to make is an amazing process. And that turns out to be what's called the temporal cell fate specification machinery. And I mean, I'm citing Chris a lot because he was my mentor, but actually he was really one of the key people that drove this forward and showed that at each division or a couple divisions, the neuroblasts cycle through a transcriptional sort of clock so that like the first daughter cell gets something called so hunchback Krupel PDM Cas, right, are transcription factors that help specify domains across that embryo I was telling you about, right, for spatial patterning. It gets reused, just like you were saying with Sonic Hedgehog in the axon versus
yeah.
dendrite. It gets reused in this temporal cell fate specification machinery. So the first daughter cell, sometimes the second, get hunchback, but then like the second and third get Krupel, and then the next get PDM Cas. And if you were, say, to make a mutation in hunchback, you would get a daughter cell that was not the right firstborn daughter cell, and then you'd go right to the Krupel positive one, and it would be so that transcriptional machinery that in the initial cellularized embryo is telling you where you are in space, in a neuroblast lineage, as those daughter cells pop off, is telling you when you are in terms of what cell you should make, right? So those are things that would have been very, very hard to figure out if you couldn't do screens and you didn't have the sort of tools available in the fly. And honestly, Chris and others were able to do it because they had defined the neuroblast lineages in the daughter cells that were made so beautifully, and it was so highly stereotyped, right? So the fact that you could go back to, if you said to me, show me the firstborn daughter cell from neuroblast 7-1 in segment 4, we could do that, right? Because we have that level of map resolution. And when you have that and you can go back and consistently look at the same cell, it's very powerful, right? So, for example, when you were looking at axons versus dendrites, probably in the mouse and the shapes, you kind of had to say, all right, this group of cells we're going to assume is probably going to look pretty similar. And if any of them change, they're going to all change the same way, right?
Yeah.
Whereas in the fly, you could say, all right, in
Yeah.
that segment, it changed this way, but it didn't in this segment. So there must be some difference, right? And so the resolution you have to ask really crisp uh, developmental questions is phenomenal in a fly. In that case, much like a worm too, right? Which has also been great for developmental studies.
So with neurons, there's all sorts of different subtypes of neurons based on their morphology, the neurotransmitters they use, and so on. And in the glial, take astrocytes, for example, not so much is known about that yet, right? I mean, do you think the complexity of the individuality of different astrocytes is a lot? Because early on, the thinking was these astrocytes are all the same.
Yep.
They're all doing the same thing.
Yeah. So that's a great question. And I may be a bit of an outlier in the field, but I'll tell you what I think. I'll tell you what other people think. There are people who might argue that the diversity of astrocytes may approach that of neurons, right? I myself think that is probably not true. And so why? Uh, First, a lot has to do with studies we've done, but there's a good reason to think it is worthwhile to program this neuron to be a little different from that neuron and a little different from that neuron because they each have to wire into a slightly different space. They have to do slightly different things. In this part of our brain, they're a little different than that, or maybe very different. They have to, some grow very locally, some grow really far. And so they have to have differences that allow them to do that.
Yeah.
With astrocytes, I believe that there's going to be subtle differences transcriptionally during development in different, and right now I'm kind of talking in mammals, right? I think there's going to be differences in different parts of the brain, like a hippocampal astrocyte's probably going to be a little different than one in cortex maybe versus amygdala, but I bet for the most part they're
Yeah.
pretty similar. And and then what happens is they're born, they migrate out to where they're going to form their spatial domains. They, as you probably know, grow and tile with one another to cover the entire parenchyma, right? And all the synaptic space. And there's also ones in the axon tracts.
And they're connected by gap junctions, right? So they
Yes.
can
Yeah.
actually, we did a lot of calcium imaging early on when Roger Chen came out with Fear 2. And so, yeah,
So
so the idea there was
just to finish
glial
the
cells
thought of
can send
diversity.
signals at long distances.
Yeah. So we can come back to that because that's really interesting. But
But,
to finish the idea of diversity,
yeah,
I think what then happens is, I'm a believer in the notion that glia evolved to make neurons much, much better at their job, right? I sort of jokingly refer to them as the helicopter parents of neurons, right? Like they clean up their messes, they kind of help shape and dress them, they give them the food and the other things they need to survive, and and essentially what they do is they try to remove some of the workload from neurons so they can focus on coding information, right? That's my belief. So I think when those sort of call maybe naive or very immature astrocytes go out, they're somewhat homogeneous with some differences across brain regions. And then once they settle into their regions, they just ask, who am I near and how do I need to service them, right?
yeah.
And then they might take on some differences in function based on who they need to interact with. But, in many cases, well, so in a mouse, it's estimated that a single astrocyte covers somewhere around 200,000 synapses, right? Almost certainly that's going to be multiple types of synapses
Yeah.
and doing very different things. And so they really need to be able to interact with a lot. So I'm in the camp which says there may be some diversity across brain regions, but I do think for most of their fundamental functions, they're going to do a lot of things in very similar ways. And there's definitely some specialized ones here or there, but I don't think their diversity necessarily approaches that of neurons. But I mean, this is my gleaning that from the literature, right? Now, then they are coupled, as you say, by gap junctions very heavily, and they seem to talk to each other intensively, right? So I actually now am starting to think of astrocytes as a giant sort of syncytium in the
Yeah.
brain that is always talking to itself. They're talking to each other, and one thing that's unclear is, how far is the reach of a single astrocyte? Like, is it only affecting the neurons within its own domain? I think probably not. It's probably doing it at a distance, right? And then the other reason I think that the initial growth is somewhat stochastic is that in the fly we can do really simple ablation studies. Like we could ablate one astrocyte, and if they were really different, you might say, okay, well that astrocyte should go to this place, and if you ablate it, that place will be
Yeah.
empty. But it's not. The neighbors just grow in and fill in that space, right?
Yeah.
And we can also in the fly, for example, track exactly the same astrocyte. So in the larval ventral nerve cord, in each half segment or hemisegment, there's 5 to 6. And for example, there's always one that is born on the dorsal part of the nervous system and migrates ventrally, and that's the only ventral one in each segment, hemisegment. And we could just uniquely label that one and say, what domain did it take? And one time it looks like this, the other time it looks like this, the other time it looks like that, right? So even if you just ask, does the same exact astrocyte always take on the same shape? The answer is no, it's stochastic. And then when you start ablating others, so I actually think what they do is they're born, they race into the neuropil, they start growing, they start to touch each other, and then once they've kind of filled in a space, they start to elaborate those fine leaflets that associate with synapses that they help make, et cetera, et cetera. So that's my sort of vision of the basics of astrocyte development. Almost certainly way more complicated, but that's where I would land.
So in the initial, these interactions between the astrocytes and neurons during development,
Mhm.
as synapses are forming, what are some of the roles that have been identified for astrocytes in determining the, I guess, initial wiring
Yeah, yeah, yeah.
during development?
Well, so there's famous studies that I would read in grasshopper where there are some glia that, and I don't know if this would actually fall into the astrocyte category, but there is one glial cell whose name is escaping right now in grasshopper where this axon is growing anteriorly and then it makes a turn right at that glial cell. And if
A
you
guidepost cell.
yeah, or
Yeah.
I can't, maybe it's a guidepost. I can't remember the name of it, but like if you ablate that, the axon just goes straight, right?
Yeah.
So, but that to me is more of that cell playing an interesting developmental role, which may or may not be conserved. The more conserved things that I think about are it's very clear from really beautiful work from Ben Barres that, so you have in mammals, neurons get born, they start to wire, and then about birth, astrocytes start to get made. And the first 30-ish days of development of mouse, several things happen. First, synapses start to form. And what Ben showed really beautifully was, if you put neurons in culture, they can form a few synapses that work. But if you provide them with the astrocyte layer, they form synapses much better, right? And then he showed you could get that from astrocyte, that effect of promoting synapse formation from astrocyte conditioned medium. And the first paper they showed on that was on thrombospondin. Christopherson, I think, was the paper where they showed one of the things that gets secreted was thrombospondin, and that was prosynaptogenic. But one of the things about the paper that was really interesting was that even though thrombospondin could help form synapses, they were postsynaptically silent, right? So it could induce structural synapse formation, but not really functional ones. And then later in Ben's lab, Nicola Allen identified things like glypicans 4 and 6, which are also secreted from astrocytes that help drive the insertion of AMPA receptors postsynaptically, right? So the fact that Ben developed this really beautiful biochemical assay that enabled them to ask what is floating in this media that does that allowed them to find many, many things. And, many of his progeny have gone on to do really exciting things. Chalo Rolu found the receptor for thrombospondin, which was alpha 2 delta in neurons, and sort of explained to us how it worked. And so there was an example of actually, because of that biochemical approach, mice were way ahead of us in flies in terms of figuring it out. So we went back and asked, is thrombospondin required in flies, we and others? And the answer is yes. There are other things like neurexin neuroligands, which Chala Arolu's lab has shown recently, not only play a role in pre and post-synaptic cells in driving synapse formation, but they're also in astrocytes. And those astrocytes play a role in helping those synapses form through neurexin neuregulin. And a former postdoc of mine had shown that that was also true in flies. So the first is synapse formation, and I think we're finally getting to the point where we have a reasonable understanding of some of the pathways. But we still have a long way to go because, to the best of my understanding, there's no one way astrocytes do this. If you find one of these molecules, then you make a knockout. You see very clear phenotypes, but they're in this part of the brain, but maybe not here, and it's a little stronger there. And so probably there are a lot of ways to do this, and it varies across brain region. But I think it's very clear now there are many factors released from astrocytes that help promote synapse formation, right?
Yeah.
Yeah, once you've
And
made those synapses, then astrocytes can secrete things like cholesterol and
yeah.
other lipids that help with efficacy and plasticity. And then in the final, well, not the final, but one of the other things that's really exciting is that it's now clear that astrocytes working along with microglia help refine circuits, right? So initially you build a circuit, you make too many synaptic connections. Those synaptic connections kind of compete to win, and then the winners survive and the losers weaken and are removed. And yeah, from C. elegans to flies to mammals, it's clear astrocytes play a really important role in that, right? And I'm
And
happy to
some of
deep into
some
those very details if you want. Yeah.
so some of the molecules produced by the astrocytes are released and maybe kind of diffuse maybe some distance, but a lot of them are associated with the cell surface.
hmm
Even growth factors, one growth factor produced by astrocytes, which is really good for neurons, is fibroblast growth factor 2,
hmm
FGF2. And when we do cell cultures, we just add it to the culture medium. But then I played around with blocking antibodies to block FGF interaction with receptor and so on. And I had these cultures that have astrocytes, but like part of the culture, there's astrocytes, part of it not.
Yeah.
And the neurons growing on the astrocytes, FGF, apparently associated with the surface of the astrocytes,
hmm
promoted outgrowth of dendrites and axons in the neuron.
Yeah.
Yeah.
Yeah, I think I mean both are possible, or both have both are clear. There are examples of both. So, in from astrocytes, thrombospondin and glypicans are pretty clearly soluble things that
Yeah.
go off. How far they go will depend on a whole variety of things. There's this one of the things you have to think about when you think about these factors is something called tortuosity of the nervous system, which is how hard is it from a thing to move from here to here? Like how many impediments are there? And there's a lot of parts of that. There's electrochemical charge, there's physical barriers, there's all that, right? So if you look at thrombospondin, it's got like not 20, but a lot of binding domains. So everything it can bind to can slow it down,
Yeah.
right? So you have to think not only about how much extracellular matrix is there? When was it secreted? Can it flow through? But then also, how does it stick to all these things? So the reach, that gets back to this reach question.
Yeah.
So thrombosponins, glypicanser would be secreted. I think neurexin, neuroligin are one of the best examples of a tethered molecule on an astrocyte that really potently affects the synapse in some pretty interesting ways. So yeah, so both are possible. And there's even some surprises, spatula 3 is or 5 is a molecule that is it looks like a secreted growth factor, right? And I had a postdoc in my lab working on this molecule, and when we looked to see how far could it actually affect the growth, so it's released by neurons and it promotes astrocyte growth. It turned out it would go not very far at all from the cell that was expressing it. So even though, and I think that's becoming true for more of these like IL like growth factors and so on, that even though we think of them floating off into space,
Right.
there's some things that keep them a bit more restricted than you might expect. So you can't assume. I think we were surprised by that. So I think the story in the end is going to be quite complicated, but that's okay. Yeah.
So during development
Mhm.
in mammals, I don't think it's so true in the worms, but in flies, my understanding it is that you get a lot of neurons that are initially born from stem cells, and they start to differentiate and interact and form synapses.
Mhm.
But then at some point, some of those percentage of those neurons that are initially generated die by
Mhm.
programmed cell death, apoptosis.
Yeah.
And then and then some of the synapses that are initially formed are removed, they're pruned.
Yep.
And you mentioned that, so Beth Stevens, I think, was the
Yeah.
one then that showed that, and she was focused, I think, mainly on microglia.
Microglia C1q, CR3,
Yeah,
yeah. But
yeah.
then also Wansuk Chung, who was a later postdoc of Ben's, worked on MEGF10, which is a molecule that actually I had found in flies in Chris's lab that was in glia that was required to eat cell corpses, but was originally found by Zheng Zhu and Bob Horvitz's lab in worms in the context of cell death, right? So, so yeah, there's, in my opinion, very deep connections between cell death and neuronal remodeling, right? So, as you say, um, especially in, well, let me start with mice, and then we can go backwards to flies. There's not as much developmental plasticity in neurons. I think you're right in worms because they're so highly stereotyped. There's like a few examples where there's some remodeling, like some synapses in a subset of neurons will go from dorsal to ventral, right? And that's an interesting reorganization. But examples where like specific synapse get clipped off are harder to find. So in mammals, it was observed a long time ago that yes, there's a huge amount of cell death in the nervous system. And if you just ask in some region of the brain how many of the cells are dying, it can range from, 15% to up to 80% of the neurons that are born will ultimately die. Now, some of those are made from stem cells and probably die quickly. But that's how a lot of people think about them, when in fact what often happens is the cells are born, they wire into circuits, they form synaptic contacts, they fire for a couple of weeks. Lynn Landmesser showed this in Chick beautifully, actually. And then half of them will just suddenly weaken, unplug, and die. And we don't know why that is. And it's a really fascinating thing. Like, it still remains a question how so clearly it's regulated by activity. People have gone on and that's one thing we're really good at is manipulating activity during development and seeing what happens. And it's very clear that there are neurons where if you have reduced activity, you'll prune and or die. There's also great examples of where if you have too much, you will die, right? So it could go both ways. We really don't yet understand how neuronal activity is translated into a signal that says prune or die, right? That's a major unanswered question that it's sort of comical we have not gotten that answer yet, right? But it's true. So then the question is, what causes them to die. There are some famous examples like dorsal root ganglion cells, right? Where these are in the dorsal root ganglia are born. They send out initially way too many projections. Their distal axon projections compete for nerve growth factor and other things. The ones that get it survive, and the ones that don't will ultimately signal back to the nucleus, and the cell fragments and dies. And so a great model for that is NGF withdrawal, right?
Yeah.
So you plate DRG neurons, you grow them in NGF, you withdraw, and then you get retrograde signaling from the axon to induce cell death, right? So that's an example. Now, what role do glia or astrocytes play in that? Um, it's everyone was really shocked when Beth found C1q, which is probably primarily microglial expressed molecule. In her initial paper, what they found, and I don't know if you remember, but they would expose neurons to astrocytes, and they would see C1q go through the roof. So they thought it was they were looking for molecules that were turned on in neurons in response to exposure to astrocytes, right? But it turns out they didn't know it. They had some contaminating microglia in those cultures, and that's where it was coming from. Because I never understood that. So I actually asked Beth, and that's what she told me she thought. So microglia make C1q, and Beth essentially showed that what is, I think, still one of the most beautiful results, and this was collaborating with Nicola Allen, who did the recordings, was in DLGN, right? So our retinal ganglion cells will wire in, they'll make too many synaptic connections, and they'll compete on postsynaptic cells for survival. And then a certain, 80% or more of the inputs will retract, and you'll have the final wiring pattern, right? And Beth found that if you knock out C1q, you don't resolve those inputs as well, right? And that raised the idea that maybe there is a positive signal from glia that somehow enables neurons to prune their connections, right? And so the two ends of the spectrum of that argument are microglia murder synapses, right? They go in and they kill, yeah. But that's almost certainly not true, right? The other is that microglia are just garbage collectors, right? The neurons make the decisions, and the astrocytes and microglia just clean things up. I actually think the answer is much more complicated than that, and I like to think of it as assisted suicide. So what probably happens is you overproject, those synapses start to compete for space. Some of them start to lose, like let's say these three. Those three, I will bet, expose a cue that says, I am competent to be pruned. And then a glial cell comes along and says, okay, finish executing your program and I will clean you up, right? They do it. And then, so it's the neurons are making the decision, but glia are required to be there for final execution, right? So that's why I would call it assisted suicide. Now, what's the evidence? One bit of evidence that that can happen at the synaptic level was Beth's observation that in the C1q mutant, you got the same amount of overwiring. And then when you knocked out C1q, when you looked at electrophysiologically from a recording from the postsynaptic cell, how many inputs are there? If glia were just trash collectors, then you would expect it still to go down to the right number, but the debris just to be there, right? But that's not what they found. They found that there were many extra functional synapses, okay, which implies that somehow C1q helps in the elimination of those weak synapses, right?
Yeah.
So now, and more recently, there's a paper from Aaron O'Shea that shows you get sort of a partial caspase activation in synapses, and that's important for them to be pruned. Now that,
Actually,
yeah,
we actually had evidence for that in the early 1990s,
yeah, yeah,
because so when a whole cell undergoes programmed cell death, then macrophages or microglia engulf it.
yep,
And this one of the signals, or the main signal maybe, is a flipping of phosphatidylserine from the inner leaflet to
mhm,
the outer leaflet. And we found when we expose cultured hippocampal neurons to apoptotic stimuli that can happen locally at synapses. And then we also had evidence that caspase cascades can be activated locally at synapses.
Yep. Yeah, yeah. So, I mean, we may get to this, but we can now look at localized activation of specific caspases with antibodies in axons that are going to
Oh,
prune, but
okay.
not others that don't. So, but you know, this all makes total sense because if you, so here's why I feel like the remodeling field has not fully embraced all of the beautiful studies on cell death that were done in C. elegans, right? So there are two papers in particular I think everyone should read. One's from Bob Horvitz's lab and another's from Michael Hengarter's. And what they asked was, does the engulfing cell play any role in cell death, right? And what they did was they took a caspase 3 partial loss of function mutant and they identified cells where they say, let's say these 10 cells are always going to die. And in the partial loss of function mutant, only 7 die and 3 survive, right? And in that background, if all you do is remove a copy of SED1, which is Draper and Fly, MEGF10 in mammals, which is in the engulfing cell, now 6 or 7 cells survive, right? That's one copy. So just removing one copy of a key engulfing receptor from the engulfing cell, now you have more cells live, right? Which provided the first evidence that I know of that the
Yeah.
engulfing cell is actually helping pushing half cell dead cells over the edge. There's also a beautiful
And
paper.
there's a threshold that suggests there's
Yeah.
a threshold. Yeah.
Yep.
Yeah.
There's also a beautiful paper. First author is Marin Teva on Purkinje cell development in mouse, where they show that in Purkinje cell development, again, you overproduce Purkinje cells, a subset of them turn on low-level caspase 3, and then microglia go up to those cells, and it looks as if they give them a little ROS, like to try reactive oxygen species to try to push them over the edge. Then they turn on high caspase 3, die, and the microglia eat them. But what's really fascinating is if you embryologically ablate microglia and you watch what happens, you know, somewhere about the same number of cells turn on low-level caspase 3, and then they shut it off and live, right? So to me, that says that you're starting
Yeah.
with the cell that's making the decision saying, here's what should happen. And the glial cell comes along and says, okay, execute, and then we'll make sure. And so I think, you know, what we need to understand is that communication if we really want to understand the role that glia are playing in remodeling. And so for example, in our lab, one of the things we've been trying to do are just identify neurons in the fly which undergo really highly stereotyped pruning. And we found one recently called the beat VAM neurons. It doesn't matter what they are, but this neuron has this big, beautiful dendritic field or axonal field. I'll just call it a neurite field that at metamorphosis prunes away completely and then it just regrows its axon anteriorly. And if we let that developmental process go and we just freeze at metamorphosis, astrocytes turn into phagocytes and they help prune all these things. We showed they eat that. If we freeze them developmentally, so they still think they're a third instar larva, using, by the way, because I know you like this, a dominant negative dysone receptor, right? So they never get the timing cue to say proceed developmentally. Then the neuron, where we've only frozen the astrocyte, doesn't prune, right?
That's cool.
So now we can go in and we can do screens and try to ask, what is the signal from the astrocyte that is telling that neuron to prune, right? And I think that is really the interesting mystery that we have to pick apart.
And okay, so what about behavioral? So flies, you can also do a lot of behavior, right?
Yep.
You can look at learning and memory, and
Mhm.
there's all sorts of motor function. You talked about olfaction. You can obviously they have sensory and motor systems, and
Yeah.
their brain is processing. So have you looked and are what's being looked at in terms of manipulating genes and selectively in astrocytes that seem to play a role in development or adult plasticity in the fly, and then look at behavior to kind of get
Hmm. Yeah.
the
So how do you
behavioral
link?
concept.
Yeah. So how can we link the developmental events to changes in behavior that are
Yeah,
specific in the adult? Yeah.
because
That's
you mentioned the helicopter Uh,
The
now,
helicopter parents. Yeah.
yeah, helicopter parents. So that's
Yeah.
what's the role for glia?
Well, so if in the fly, actually, I don't know that we've made good progress on that. There's I could tell you about how astrocytes play important roles in behavior, but those are more neuromodulatory roles rather than
Oh,
developmental ones. But if you were
okay.
to look, for example, in mouse at C1q, right? Those so that molecule is just sort of popping up everywhere in the nervous system. And if you were to look at maintenance, right? So one of the interesting papers that came out of Beth's lab, well, what the first thing was that they showed that it is indeed microglia through CR3 that are doing the engulfing events. And then Wansuk Chung showed that that's probably redundant to some extent with astrocytes using MEG-F10 and MERTK. If you look at the adult brain, what Beth looked at was a disease model, right? One of the Alzheimer's models where synapse loss occurs in a kind of progression throughout the brain. Um, and what she first noticed was if you look at how synapse loss occurs in these different brain regions, about a month before they start to lose synapses, they start to get coated with C1q, right? And the order of that appearing in the brain is the same as the order of synapse loss. And if they knocked it out, they got a partial suppression of synapse loss in that model, right? So that's not necessarily like, I think what you're asking is, can you give me examples of things that are used for plasticity during development that are required, for example, for learning and memory or plasticity? That's harder. I don't know that we have those yet,
Yeah.
but I would argue that's because we know so little about remodeling right now that it's hard to do that, right? I think that's going to happen,
Yeah.
right?
Okay,
Just
let's go to the adult brain and how the astrocytes modulate the neural function and
sure,
vice versa. And
sure. Yeah,
um, you know, there was, and I didn't read word for word all three of these papers, but very recently in the May issue, one of the May issues of Science, there were three papers including one from your lab, three studies that all focused on a particular neurotransmitter or yeah, essentially norepinephrine
yeah,
and the interesting roles it's playing in and that did include behavioral analyses, at least
yeah,
in some of them. So can you kind of summarize
sure.
those and your contribution to that?
Hmm. Yeah. So in the adult brain, one of the reasons I sort of jokingly call glia helicopter parents is because their whole life is about getting neurons what they need. And so that includes things, one of the most famous roles for astrocytes is mopping up neurotransmitters like glutamate and GABA through these channels that they express, delivering nutrients like glucose and or lactate, removing metabolic intermediates and providing for gas exchange through interactions with the vasculature. So this is all, kind of classic housekeeping work that's going on the astrocytes are doing. And some people sort of say, well, hey, that's kind of boring, but I think maybe it's boring, but if you didn't do it, they didn't do it, you'd be dead, right? Your brain would not work. And the other is that we don't know this yet, but I bet every one of those mechanisms provides another opportunity for astrocytes to regulate neuronal activity. So, for example, how might an astrocyte change glutamate tone in the brain? Just take it up a little slower,
Yeah.
right? Or how might it slow a neuron down for firing or speed it up? Give it more or less energy, right? I bet in the future what we'll find is each one of those metabolic roles are also ways in which astrocytes contributed to regulating neuronal function. Now, the one that you brought up that we have spent our most time on is signaling, adrenergic signaling, I'll call it. So norepinephrine in mammals, octopamine, tyramine in flies. But my friend Scott Waddle would warn me, it's not totally fair to call it adrenergic because there's some reasons to believe there are some subtle differences in which species have octopamine, tyramine, and norepinephrine. However, for the most part, octopamine and tyramine act like norepinephrine and epinephrine in mammals. in the fly. So there was this. So when things like GCamp came out, and really good, calcium indicators, people realized, and it had been long thought that astrocytes don't uh, fire or respond to neural activity in really dramatic ways with their own signaling events. But then a number of groups realized that, in fact, if you look, you could, with GCamp or with Flora or other things, you could see these really interesting patterns of calcium signals
Yeah.
in astrocytes. And so it raised this idea that, oh, well, maybe that is activity, right, in an astrocyte, and that represents some signaling event that allows them to respond to neuronal transmission and maybe in turn around and regulate it, right? And so, and actually, they're in the first 10 years of my lab. Uh, this was a very hotly debated topic at glia meetings. In fact, like arguments broke out constantly where you would have, one camp which would say it's absolutely crucial and potently regulates neural activity, and another one would say it does nothing, right? And a lot of experiments were done using what I'm going to call sort of brute force methods, which I don't mean to sound like disparaging. It's just that this is what we had. So you inject a cell with EGTA, like an astrocyte, and then an event that should happen is altered. That tells you
That's
something, right? But
crazy.
I don't know. I don't know exactly what that tells you. So,
So EGTA is a calcium chelator, so that's a
yes,
very
sir.
crude experiment, stopping up.
Right. So
Okay.
you sop up all the calcium and you see an effect.
Yeah.
And then other things were done like channel rhodopsin to create, big pores in the hole and the membranes and change calcium signaling. DREADs, designer
Hmm.
receptors that you can activate could lead to interesting changes in calcium signaling. And then there was one made by Balcock, which used to be called CalX. It's essentially a calcium uniporter or importer that was turned inside out, so it pumps calcium out, right? So all of these approaches, when used in animals, supported the notion that if you perturb astrocyte calcium signaling, there were effects, real effects on circuits, right? But what we still didn't know was what are the endogenous molecules that astrocytes would use to maybe receive neurotransmission signals? What were they responsive to? And then if they were responding, did they, in the end, use that information to change downstream behavior? So we got interested in this when a postdoc came to my lab named Jiguo Ma. Uh, he came, I think, in about 2010, and he said, "I want to study calcium, signaling in astrocytes in the fly." And honestly, I said, that field is so hotly debated right now. I'm not sure you want to get into it. But to his credit, he said, "We can figure it out." And I was like, "Okay, let's go." And in what he did, and we published this back in 2016, was he showed that. So he developed this preparation where he could pull out a larval brain from a fly and image it and watch astrocyte calcium signaling. And then what he did was he noticed there were two types. Just like in mammals, there are these little microdomain flashes that occur in the fine processes of astrocytes. And then there are these whole cell fluctuations. And in his case, the whole network, those whole 180 group of astrocytes in the larval brain would kind of rhythmically go up and down together as if they were talking to each other, right? So then he added TTX to block neurotransmission, and he found that those little microdomain transients kind of kept flashing, most of them, right? But what he lost was that huge oscillation, right? And what he later figured out was there are neurons in the larval brain called TDC2 neurons, which make octopamine and tyramine. So you can think of those as the equivalent of the mouse locus ceruleus neurons, the adrenergic neurons. And in this preparation, for some reason, they rhythmically fire. And what he saw was when they were bursting, astrocyte calcium would go up in the whole cell, and when they stopped, it would go down. And when it went up, when they fired, it would go up. And if you silenced them, it was flat, right? So then in the presence of TTX, he started bath applying GABA, glutamate, acetylcholine, dopamine, no response, but octopamine and tyramine fly norepinephrine, he got a beautiful response. Then he did a screen. He figured out what receptor it was. It was one called the octopamine-tyramine receptor, which is an adrenergic receptor in the fly that's expressed on the astrocyte. And in a paper in 2016, he showed that the TDC2 neurons basically fire, release octopamine-tyramine that binds to this receptor on the astrocyte. Then the astrocyte, through a trip channel called water witch, generates this calcium wave,
Hmm.
right? And ultimately,
So,
he was.
so that calcium channel in the membrane, the trip channel.
Hmm?
Okay, so the octopamine receptor, adrenergic, if you will, or not. So they're typically coupled to cyclic AMP.
Yes. Yeah, yeah.
So are you getting, is it a phosphorylation that of the trip channel that cause it to open or?
We don't think so. But but
Oh,
but cyclic AMP is involved, and let's
okay.
come back to that. So
Okay.
so for this first paper, all Jiguo showed was essentially norepinephrine is released, it binds the astrocyte, it generates a calcium signal, and then that calcium signal can silence downstream dopaminergic neurons somehow, right? And he was able to show that if you just activate a non-endogenous trip channel in the astrocyte, that was actually also sufficient to
Uh,
silence those dopamine neurons. So that astrocyte calcium signal is sufficient to silence those DA neurons. And then he showed that it was actually through an adenosine receptor, right? Okay. And then he showed it was important for two behaviors, olfaction and a startle-induced escape response. And then we published that. And so that was nice because it showed the octopamine-tyramine receptor, that adrenergic receptor, was required to activate this calcium signal that could silence DA neurons through the trip channel, and it was important for both physiology and behavior. And in this recent set of papers, in the much more complicated nervous system of the mouse and the zebrafish, the Papuan group and the zebrafish group of Misha Ahrens at Janelia found it essentially the same pathway is present in both zebrafish and mouse, right? So they did really beautiful work showing that astrocytes express adrenergic receptors and they can respond to norepinephrine. Now, the notion that astrocytes responded to norepinephrine was well known in the field, and probably the person who deserves the most credit for that is Dwight Burgles. The first talk I ever heard that showed that was at a Gordon conference where Dwight presented that, and he essentially he was trying to figure out what generated calcium waves, and he had a head fixed mouse on a treadmill, and they would provide light input and look in visual cortex and at astrocytes, and they could never figure it out. And then they actually figured out that when they would shut off the treadmill and go to lunch and turn it back on, and the mouse got startled, there was this huge wave of calcium. And they eventually figured out that that was a startle-induced release of norepinephrine, and that was what was causing it. And Dwight, so that was a really beautiful paper from Dwight. And then that sort of got us all thinking, okay, we got to think about norepinephrine. Now, Misha's group and Tomas's group showed they identified the receptors in the mouse and the zebrafish that were expressed in the astrocytes, showed those generated the calcium wave. And in the case of the zebrafish, and probably also the mouse, that somehow leads to probably ATP release, which is broken down into adenosine and then signals through A1 adenosine receptors on the neurons, right? And in Misha's case, in zebrafish, they showed it was very important for this really interesting behavior they study, which is like a learned helplessness behavior where they make the fish swim in this virtual environment and they basically make it think it's failing over and over, and eventually it learns helplessness and it stops swimming. That requires this signaling through astrocytes for them to make that decision. And then what Thomas's group showed in his case was that synaptic depression in hippocampus was required the same signaling pathway, right? So it was neuromodulators, norepinephrine binding to astrocytes, them releasing ATP, it being broken down to adenosine. And then you get activation of the A1 receptors on the neurons. And so that was really exciting. And I think what it says is that the fact that it's in a fly, it's in a zebrafish, it's in a mouse, says it's a very old pathway, right?
Yeah.
And really important. And so that's kind of what their papers did was really bring that to mammals. And now what's so exciting is that the field has the tools to go back and ask, when you knock out those receptors, specifically in astrocytes, how do they affect the sort of interesting behaviors that we can, or
Yeah.
electrophysiology in a mouse? So that's going to lead to really exciting things.
I
Now,
was I was thinking of sleep, right?
oh
Adenosine,
yeah, yep.
like my understanding is throughout the day, adenosine levels kind of increase, and it's like it promotes sleep, and caffeine promotes alertness
Hmm.
by inhibiting adenosine's action. So it
Yeah.
would be interesting to look at sleep in the flies, right?
Well, so there are already deep connections to astrocytes and sleep. And Phil Hayden, I think, was one of the people who led the way on
Oh,
this
yeah.
and has been studying that for a very long time. But over and over, sleep and rhythmicity have been shown to be importantly regulated by astrocytes. In fact, one of the most beautiful papers that convinced me first in flies that astrocytes were doing something really interesting was by Rob Jackson at Tufts, who showed there's this molecule called ebony that processes some neurotransmitters that is rhythmically expressed in astrocytes in the fly and is very important for circadian rhythmicity and sleep, right? And he showed that really, it was a neuron or a cell paper long ago that behavioral change mapped very clearly to astrocytes, right?
Hmm.
So our paper that came out with this group went a step further in terms of asking what else can astrocytes do and how much does that change? So as I told you, Dwight had this really interesting experiment where he had a mouse on a treadmill and he was trying to figure out what had generated these calcium signals in astrocytes, and he found that it was a startle response. If, for example, separately, he looked at visual system astrocytes and gave them light input, there was no response. But if he coupled the light input with a startle, he got a super response, which was way bigger than the startle response alone. And so he suggested in a paper, I'm 5 or 6 years ago, that norepinephrine release makes astrocytes more sensitive to local circuitry, right?
Huh.
And that was really interesting, and that inspired Kevin Gutenplan, who was in my lab, to sort of ask, what happens, how plastic is the response of an astrocyte to any given neurotransmitter? So, as I said, if you take a larval brain, you put it in a dish, and you put TTX there, and you have GCAMP, so a calcium indicator just in astrocytes, and you add GABA, glutamate, dopamine, acetylcholine, no response. If you had octopamine or tyramine, response. What he found was if you pre-treat the astrocytes for just 3 minutes with octopamine or tyramine, so norepinephrine, and then you add, GABA, glutamate, acetylcholine, now they respond to everything, right? So
Wow. Yeah.
pre-exposure of an astrocyte to an arousal cue, norepinephrine or adrenergic signaling, takes them from not listening. And again, this is a model, right? So in the actual brain, there's probably always varying levels of norepinephrine, and this is all on a graded scale. But if you just use this prep, if they don't have norepinephrine, they don't respond to these other neurotransmitters. If you add it, now they do. And what Kevin showed in that paper, you had asked about cyclic AMP signaling, right? When you add octopamine or tyramine, fly norepinephrine to an astrocyte, the octopamine tyramine receptor leads to that calcium signaling event, but that's through phospholipase C beta. But what it also does is through GPCR signaling and changes in cyclic AMP level is reduced levels of cyclic AMP. And what he showed in the case of the dopamine response is that reduction in cyclic AMP leads to the blockade of internalization of a dopamine receptor. So these astrocytes have these dopamine dope2R receptors, and they're always just internalizing them. But when you stimulate the octopamine tyramine receptor, they block that, and now they accumulate on the surface. And now the cell can respond to dopamine, right? So the pre-treatment with an arousal cue lets that astrocyte go from not responding to dopamine to now responding. And he showed that the same mechanism was true in mouse, or actually rat cultured astrocytes. And there he was actually able to show that using an antibody to the extracellular domain of DRD2, which is the ortholog in mouse, that you do actually see accumulation after blockade of that internalization pathway on the surface and responsiveness. Now,
So this suggests a role for astrocyte. So we remember things that are startling.
yeah, yeah.
Like you watch a horror movie and you go along, and then all of a sudden, whatever,
Hmm.
something bad happens. Then if you talk with someone about the movie a
Yeah.
year later, and you focus on that.
Right. So, not just startle though, but right, arousal and attention. That's really
Yeah,
driven by norepinephrine,
yeah,
right? And so that's the idea, is that this arousal cue makes astrocytes go from not really listening to other synapses to
yeah.
now listening to everything, right? And it turns out that in that paper, he also showed they respond to glutamate, GABA, and acetylcholine. But whereas dopamine is modulated by this slow reduction in cyclic AMP, uh, acetylcholine, well, actually, I think we can say GABA and glutamate so far are instantaneously able to activate an astrocyte if they're in the presence of octopamine, tyramine, or norepinephrine. So there, it's more of a coincidence detection. And
Hmm.
I find that interesting because, acetylcholine, GABA, glutamate are these fast-acting neurotransmitters you'd want to adapt to very quickly. Dopamine's more of a modulator that maybe you want to have a slower response to. I don't know if that's meaningful, the differences in the
Yeah,
timing, but it was fascinating. And then the most important thing, obviously, Kevin did was to go on and show, at least for dopamine-driven behaviors, that this astrocyte signaling pathway was critical for example, a larval writing behavior, where you take a larva and you roll it over onto its back and it gets startled. So you get we believe, a burst of octopamine tyramine, and then dopamine drives the motor behavior. And we could essentially make that happen faster or slower by modulating this signaling pathway just in astrocytes. So what you're implying is that as we look at more sophisticated things like
yeah.
learning and memory, there's probably going to be an important role for astrocytes. I totally agree. And I think the exciting thing now is that, with Tomas and Misha's work, people can do that in systems where you can do really beautiful context-dependent learning and memory, and you can couple that with really outstanding physiology, right? It's still harder for us in flies to do the level of high-quality physiology you could do in a slice prep, but now that's going to be possible, and I think the roles are really going to expand.
Now, to finish up, yeah, there's not going to really be time to talk too much about injury and disease. I was
Oh,
going
sure.
to, but it's would taken up a lot of time, maybe a different time. Um,
Sure.
so
You could get me to blather on for hours about axon injury
yeah, I
and
know you've done work on that, and it's intriguing. But there are definitely important roles for glial cells, astrocytes, and
yes.
a lot of it's been studied in the peripheral nervous system, though, where you can get regeneration of axons.
Hmm.
Less than. What about in the fly? If you completely, did you say this and I missed it? If you completely knock out the glia, when does the animal develop, nervous brain develop at all? Or if it does, what's wrong with it?
Uh, so if you ablate, well, so the best experiment for that is this old mutant. I'll tell you the name of the gene and you can guess the phenotype. It's called glial cells missing.
Okay.
So this mutant was fascinating. It was found in a Yoshiki Hoda's lab, as well as Corey Goodman's, and there are a couple of really exciting cell papers. In a glial cells, so GCM is a transcription factor. It comes on, is the earliest marker for a cell in a fly that's going to become a glial cell. And if you knock it out, cells that should have become glia completely transform into neurons. And then if you overexpress it, a bunch of neurons now turn into glia. So that's like a great tool. Now it turns out probably to be something of an evolutionary uh, oddity of the fly that is used in the embryonic nervous system because they have to make glia really, really fast. But in that animal, there's no glia. And what happens is the animal dies as an embryo, and macrophages just start attacking the CNS because there's no sheath isolating it. There's no nutritive support. Like there's no role for
Okay.
glia.
Yeah.
And in fact, if all you do in a fly is ablate astrocytes, which are probably on a per hemisegment or half segment basis, 5 to 6 out of the 35 glial cells, they also die as late embryos, right? So you got to have your data.
That's that's
Yeah.
a
Yeah.
yeah.
In fact, we even did one of the ways we convinced ourselves that astrocytes are important for synapse formation in flies is we let the animals. So in a metamorphosis, you break down the whole larval nervous system and then you then you build the adult one. And it's about a 4-day procedure, metamorphosis. The first couple days are breaking things down and wiring, and the last 2 days are forming circuits. So at midpoint, 48 hours into metamorphosis, if you look by EM, there's almost no synapses in the brain. So we ablated astrocytes at that point and then asked what happens to synapses. And what we found was a dramatic decrease in synapses in those brains. If you ablate all astrocytes, the animals will die. But you could kind of, do a dose response where you get down to a very small number and then you could see those changes. So
Interesting.
there's, yeah, it's it's really hard to get a nervous system without any glia in there, right? Because complex nervous systems have just evolved such that they can't do without them, right? There's so many essential functions. Now, there are examples that are a bit striking where, for example, if you eliminate microglia by a couple different methods in a mouse, everyone was kind of shocked they don't fall over dead, right? But I'm not particularly shocked by that because you can make a nude mouse, right, that has no immune
Yeah.
system and it's fine, right?
Yeah.
And also, I think that in that case, astrocytes are taking up a lot of the slack, right, for remodeling and other things.
So I think
So,
your work and others that are working on the glia is going to be very important for translational research
yeah,
at some point. We did a lot of work on the effects of exercise, intermittent fasting,
yeah. Mhm.
and I'm sure there's some role for the glial cells and
Yeah.
the beneficial effects of that.
They are, I believe, the nutritive cell of the brain, right? Like they are the way that everything kind of gets in and out of the nervous system. And
Yeah.
if it's important, there's somewhere along the way there. I don't know of good examples, and I might have been I just missed this, but of good examples yet of how that will work, but I'll be shocked if that's not the case, right?
Yeah. Yeah.
They just play so many crucial roles in making sure the neurons have what they need and they get rid of what they need to get rid of that it's
Yeah.
got to be there. So,
Yeah.
yeah.
Okay. Well, thanks a lot, Mark, for
Yeah,
taking
yeah,
the
my
time. I
pleasure.
know you're busy. You got a big active lab there in
Yep.
Portland, and
Well, I want
that's
to thank
great.
you.
I'm
I
glad I
want to
had
thank
the
you, Mark.
opportunity to read some of your work and kind of keep up to snuff on what's going on in the field.
Well, it's my pleasure, and I want to thank you for doing this because I think, especially now more than ever, we scientists need to do a better job of getting out there and explaining to people what we spend our days doing
Yeah.
because, we're in a bit of a precarious moment for science. So again, thank you for making this stuff happen.
Well, everything you're doing, and I did, maybe this too, is to benefit the public, and it will.
Yeah. Yeah, I'm confident in that, but we just got to do a better job of showing them the goods.
Yeah. Okay, thanks. Say hi to Nate.
All right. Thanks, Mark. Take
Yeah.
care.
Bye.